Really? That's bullshit. I'm going to text them.
The one at the, we can't get the one up there. The one up there is bio. Yeah, no, we can't have their shirts though.
Okay, I texted them. We'll see. Maybe we can get free ice cream after this. So, welcome back, everyone, to hopefully this is the last class ever for juniors anyway. The last class of the year. Seniors, 3 people were seniors sitting over there before. It's the last class ever, maybe. Maybe they already had their last class ever and they didn't come today. Pretty good idea. So, okay, I just want to review briefly what we talked about last time and then tell you more fun stuff this time. So, to jog your memory, last time we were here, we talked about the motivations of trying to understand why one would care about connectome coding. We talked about applications in biomedicine and psychiatry in particular, and artificial intelligence. Then I told you a little bit about why it's hard, in particular that there are a lot of different possible brain networks, and even super trivial stuff that you guys think you know how to do, like take an average. It's really not that obvious how to do in these settings. So before I start new stuff, does anyone have any questions about any of that stuff? Is it all super clear? Everyone's happy? Okay, so today I want to start by talking about different statistical models. So you guys, everyone has taken a stats class, so you know about statistical models, such as, for example, a Bernoulli is a statistical model with a one-dimensional parameter p. It's just a probability of a heads or a tails, or in network data, it's a probability of an edge, right? That's a very simple model. On the other hand, so this is number of parameters, and this guy has 1. On the other hand, if you recall from last time, there's 2 to the n squared possible different graphs, networks, if the network has n vertices. So I want, on this side of this line, to write down what is the most complicated possible statistical model. Right? So in this case, what a Bernoulli model says for the perspective of networks and connectomes is that every edge is independent and every edge is sampled with exactly the same probability, and that is probability p. So that means the probability of connection between left hemisphere and right hemisphere is the same as like auditory cortex to prefrontal cortex to whatever else. I don't know what other anatomy things you know. But that's what that would mean. Every edge is independent and they all have the same probability. In random graph land, that happens to be called an Erdos-Renyi random graph, which just means, as I said, every edge is independent and sampled from the same probability. So what's the most complicated possible distribution of networks when you have n nodes? Anyone know, like, what would it do? What would it say? Any ideas? Okay, why don't we go the other way? What's the next simplest model, statistical model of a network? This model, it just has a single parameter. Let's see if we can think of a model that has, say, 2 parameters rather than just 1 parameter. Binomial. That's the distribution, so I'm happy about that. Binomial really only has 1 parameter also. It's the total number of, say, heads you get if you flip the coin n times. Remember, we have this network. I'll just draw it as an adjacency matrix. There's going to be some 1s and some 0s in some places. And so the 1 parameter model says every single edge is sampled with exactly the same probability. They're all independent. So let's make up a slightly more complicated model. So I'll give you a hint. What I'm saying here is Aij is sampled with, according to a Bernoulli, with probability P, and I'm going to write independent and identical, which means each edge is independent from all the other edge, and they're all sampled from the identical distribution, meaning Bernoulli with probability P. So based on that, can you think of a slightly more complicated model? Let me try to motivate it for you. Let's say we're trying to differentiate between people that are schizophrenic and people that are not schizophrenic. And we have two cohorts, we measured their brains, and we estimate their connectivity, and then we're going to try to build a statistical test to see if we can differentiate between these two populations, and we're going to assume an Erder-Shreni model. So what we're going to do is estimate the probability of an edge in every single subject from one cohort, and look at the probability of an edge in every single subject in the other cohort, and see if they're different. That might work if, for example, being schizophrenic means you have more or fewer connections than normals. But what if we're comparing, say, schizophrenia and depression, or people who are really creative with people who, or say people who are really good at music versus people who are really good at math? A priori, it doesn't feel like one parameter, just the probability of a connection, is going to be sufficient, right? It's not probably going to differentiate. The differences are going to be more nuanced than that. So we want to build more complicated models, more sophisticated models that have a greater ability to differentiate between individuals and populations. So let's make one up. What do you guys want to do? No takers? These are two assumptions. Does anybody want to propose an idea that deals with one of those assumptions? Yeah?
Well, assuming that the connections aren't independent, another parameter might be like the degree of each vertex that affects the probability.
Yeah, okay. So the proposal, if I understand it correctly, is to relax independence and say somehow these edges aren't all independent. So maybe they all have some probability P, but there's also like, say, some correlation parameter rho that says, they're actually quite correlated with each other. So if there's, bless you, if there's an edge here, there's more likely to be an edge here because they're correlated. That's a really natural way. You relax this independent assumption, you add another parameter, and that is great because now we could characterize individuals with this two-parameter model. And that's more flexibility. It's still pretty easy to estimate, just do parameters. What else can we do? So that idea was relaxing the independence one. What if we relax the identical one? How would you want to do that? So let me ask you this. Do you believe that the probability of a connection, say, from primary visual cortex to secondary visual cortex within the same hemisphere is as likely as a connection from, say, visual cortex in the left hemisphere to motor cortex in the right hemisphere? Not the same. Okay, so let's write down a model, a statistical model that has that property. How do we do that? Say, this is left V1, this is left V2, this is right V1, right V2, and the same, those are the rows, the same on the columns. What would we do? Yeah?
You could assign weights to all the connections.
Yeah, so if I understand correctly, each potential connection could have its own probability. Right? Who's to say that need to be identical? So that rather than saying Bernoulli P, we can say Bernoulli P sub ij. So the probability of a connection between I and j is one thing, and between, say, I and j prime is another thing, and I prime and j is another thing, etc. But if we did that, we relax that, then we would have n squared parameters, right, for n vertices, because there's one parameter for every potential edge. And that's called the heterogeneous heterogeneous heterostrainy graph. It's another very commonly studied statistical network where just every edge is independent and every edge has a different probability. Are you guys with me? Does anyone have any questions about this? Is anyone totally lost? You guys just quiet? You're very quiet. It's a little scary. Okay, so that's every single edge has its own probability. This is every single edge has the same probability. Can we think of anything in the middle? Right, because that's a lot of parameters. N squared. How many regions are there in your brain? Someone shout out an answer. Yeah.
Can I ask a question first? So this n squared, does that include like the matrix? I mean the parameter for saying that they're not independent, like the correlation?
No. That's literally, so we're just saying this. The probability of a connection between neuron I or node I and node J is Pij. So for every potential connection, there's a probability. There's no correlations. That's a good question. So how can we get a different number? So someone was going to tell me how many regions there are in the brain. Does anybody have a guess? Just throw out any number you want. 200? It's a great number. Does anyone know why 200 is such a good number? It's even. It's one nice property. It's a multiple of 100. I really like 100. So like if your brain was symmetric, there could be 100 regions on one side, another 100 regions on the other side. That would make sense. Does anybody have another guess? Come on. I'll give you a hint. It's an integer. Say some integers. I promise you it's not such a bad guess. Come on, I'm just going to wait. I know a lot of integers, so anyone you say will be fine with me. You must know some. I recommend even integers, if that helps. 100 is another fantastic guess. That would be, say, 50 on the left hemisphere, 50 on the right hemisphere. Totally reasonable guess. Any other guesses? I can give you more hints. It's positive. Positive even integers, basically what I'm looking for. Does anyone think way more than 200, like 2,000? Do you think 2,000? Do you think 5,000? Anybody think 10,000? How about, anybody think a million? Yeah, a million?
2 million.
2 million. That's a good number. Anyone think 2? Does everyone think the answer is definitely not 2? Raise your hand if you think the answer is definitely not 2. Raise your hand if you think the answer is definitely not 2 million. That's cool. So, yeah, no one knows. Your guesses are just as good as anyone else's, as far as I can tell. Neuroanatomists fight about this all the time. They take these images of the brains, and this dude is like, no, this region ends over here. And then the other dude says, no, it ends like a little bit over to the left. And then they fight about it for years. And what are they fighting over even? I mean, the brain is continuous. Things change kind of slowly over space, and things are intermingled. And it's not like each neuron has its own job. It's not like someone built a computer and this is the CPU and this is the hard drive. It's not. We're biological organisms. There's no right answer. So everyone who said a particular number is right. Everyone said who didn't say a number is wrong. So good job for people who said a number. But it's related to a question I asked at the end of class. I don't know if any of you figured out the answer. How many neurons are in your brain? Yeah? 5 million? And just for clarification, what species are you? Human, okay. That's a guess. Does anyone have a different guess for human? 100 billion? That is a quite different guess. And you're also human. Is that right? No? Are you at least in the genus Homo? I'm gonna write Homo sapien. And then if you're some subspecies or whatever, you let us know. Okay. How would you count? Counting to 5 million is going to be really boring. And counting to 100 billion, you're going to die in the middle. Right? You just don't have that many seconds. So how would you estimate the number of neurons? Forgetting number of regions, just number of neurons, yeah?
Well, you can measure the total volume, and you can measure the density, and you can multiply the density by the volume of the brain.
That's a great idea in theory. In theory, communism works. So let's unpack that a little bit. How big is a human brain in volume? Yeah, about that big. Let's say 1,000 volume units. Who cares what the right answer is? It's like the size of a grapefruit, maybe. Now you're going to count the density of what? Cells?
Sure.
Okay. Are you going to count the density of cells in the whole brain?
No, in some small, take a small sliver.
Okay, let's take, say, that dot. Okay, there's 100 in that dot. And then multiply that 100 by the number of dots that size in the brain. Okay, it's a great idea. It's what lots of people have tried. What are the problems with that idea?
Cell size varies.
Cell size varies. Any other problems?
Doesn't capture like gray and white matter distribution.
It doesn't capture gray or white matter distribution, and like the cell density will change whether you're in gray matter or white matter. So yes, it turns out that this was the best idea that people had for about 100 years, but the variability of cell density within a local volume is hugely variable. So like, if this is your cerebellum, then individual cells or granule cells are tiny and they're super packed. And if this is like somewhere in your white matter, there's almost no cells. And so it's really hard to address that. You need to do a lot of different dots in a lot of different places and have a lot of information about where things are dense and where things are not dense. So what's another idea? You got neurons. What do all neurons have? What's the brain of a neuron? Or the brain of a cell? Nucleus, yeah. And what's the brain of the nucleus? What? Nucleolus. And what's in that nucleolus of every single cell in the world? DNA. How much? How much? A bunch?
All of it.
All of it. It's true. All of your DNA is in. Yeah, it turns out every single cell in your body, and basically in all human bodies, has exactly, pretty much the exact same amount of DNA. Right? You've got 6 billion base pairs, and they're all in there in every cell. So, based on that, how are you going to do it? You know something that's preserved. Yeah?
Precipitate all the DNA?
Precipitate the DNA, that sounds complicated. Is that like evaporate it in some lab? I do computational stuff.
Me too.
Okay, it sounds good. If maybe, right? I don't know what it means though. If anyone can say it in words that I know. Yeah?
I think what she means is essentially you can melt all this brain down and purify out the DNA.
Uh-huh.
And if you know the weight of DNA in one cell, now you have all the DNA in the whole brain, you can maybe find that out.
Okay, so I think if I understand what he's saying correctly, he's saying take a human brain, ideally from a dead human, but I don't know him, and melt it. I don't know how you melt brains, but presumably you can melt the brain. Oh, maybe in, did anyone see Thor Ragnarok? So do you remember that dude had the melting stick? Remember the melting stick from that movie? You just use that thing on the brain and melt it. Does that preserve DNA, the melting stick? Yes?
I mean, I think he means biochemically lyse it.
Oh, biochemically lyse it. But doesn't the melting stick do that?
Less elegantly.
I thought it was a cool stick. Okay, so you biochemically lyse all the cells, you get all the DNA out, you kind of know how much DNA weighs, so you like weigh it, and then divide by whatever. Yeah. So that's the way that people did it more recently. I don't know if they had a melting stick, but it's something like that. And the answer was pretty close to 100 billion. It was like 85 billion plus or minus a billion, basically. So whoever said about 100 billion was pretty close. Whoever said about 5 million, I'm pretty sure you're a mouse. Because that's about how many cells, neurons in a mouse. So just think about it. Maybe that's your spirit animal or whatever. Okay. So 100 billion cells, that's cool. I asked you how many regions. That's going to be important because we're not going to build up a 100 billion node network from a human brain because I don't have a melting stick and I can't estimate all the stuff, right? So we're going to partition the brain into regions. And we get to choose a number of regions because we're engineers and we choose things like that based on preferences and models and how much data we have and stuff like that. So maybe we'll choose 100 because 100 is a really nice round number, which means we're going to partition the brain into 100 different, like, spatially co-localized thingies and go about our business. And that's what lots of people do. So we now have 100 regions, which means we have 100 squared parameters in this model. Right? And with 100 squared parameters. 100 squared is 10,000. It's cool, you guys can do that. So how many data samples do you think you would want in order to estimate a 10,000 dimensional parameter? Let me give you a hint. Let's say I have 2 points in 3D, a point here and a point here. And I want to fit a plane through them. How many different planes will go through these 2 points? How many? An infinite, right? Because this line is restricted. The line is in 2D, but we can rotate it around the axis of the line and at any angle, and it's still a plane that goes through those 2 points. Okay, so if you have n points in d dimensions and d is larger than n, there's an infinite number of ways to get the right answer, to have a good fit. Right? This is what's overfitting. You guys have heard of overfitting maybe in machine learning or whatever. That's what overfitting is. You have essentially more dimensions to fit than samples you have. So we just said the brain has 100 regions, so there's 10,000 parameters to estimate. Now we're going to do a study. We're going to get IRB approval and write a grant. And this grant's going to be great. It's going to be like $1 million. And we're going to study and we're going to put how many humans' brains and get MRI scans to be able to estimate the networks in this study that we're conducting. You guys have read studies. How many samples are in those studies? Just in any study? 20. 20 is a great study, right? It's like most studies have about 20 samples. So okay, we have 20 samples and a 10,000 dimensional parameter. How are you guys feeling about that? Not that great? You should tell that to people who write neuroscience imaging studies because they don't know this as far as I can tell, which is why they keep having studies with 20 people and estimating 10,000 dimensional parameters. And arguably that's a big cause of the reproducibility crisis. But anyway, it's not going to work. If you have 20 samples and 10,000 dimensions, we're not going to be able to estimate that. So now we as a team need to figure out a model of these brain networks that we can estimate. This one parameter model is going to be sucky, right? Because it probably is not going to have enough differential power. This 10,000 dimensional model is also sucky because we don't have enough samples to estimate it. So we need something else. So somebody's going to make something else up. Any other ideas? Who said 2 regions before? Someone said maybe there's just 2 regions. You're not owning up. I think it's a great idea. Because I can divide the brain into, say, left and right hemisphere. left, right, left, right. Okay, and now I can say maybe there's some probability P of connections within the left hemisphere and some probability P connections within the right hemisphere and some probability Q from left to right and right to left. Now we have two parameters. And this is actually not the worst thing ever. Like if you look at a human brain network, or really any bilateral animal's brain network, it is typically the case that there's way more connections within a hemisphere than across a hemisphere. That should be kind of a natural thing to happen because they're closer, and to get all the way to the other hemisphere, you need a really long axon, and that's energetically expensive. So it really is often the case that, to a first approximation, you can say that within hemisphere connections are bigger than across hemisphere connections, so we would just write P is bigger than Q. And now we have a two-parameter model. I'm going to call that an affinity model. Because if you're in the same group, you're likely to make a friend in that group. If you're not in the same group, then you're less likely. Okay. Now, that's a pretty big hint. Can you guys think of, say, a three-parameter model? Yeah?
If you assume that the probability of crossing between the two lobes is different than the probability of staying in the left lobe, it's different from the probability of just staying in the right lobe.
So you want like a PL and a PR?
Yeah.
Cool. Three-parameter model. That was easy. I'm going to call that a symmetric stochastic block model. It's symmetric because this and this are the same thing. It's a stochastic block model. It's stochastic because the probability of the edges are stochastic. It's a block model because there's way more nodes than two, but we've basically said there's two groups, and if you look at the probability matrix, it's in a block structure. So as a symmetric stochastic block model. Okay, let's do 4. How are you going to get a 4-parameter model? Someone is saying the answer should do a Jedi mind trick and just force you guys to say it. You know it, right? So we could just make this a Q right, left, and a Q left, right. Now there's 4 parameters. I'm assuming you guys knew that one. Okay, great. That's 4. That one's called just the SBM, the stochastic block model. Okay. Now 4, we had 20 samples, so we can do more than 4. How will we make this even more general? More parameters. Yeah?
We could do rough regional differentiation like neocortex, cortex, cerebellum, brainstem.
Sure. We could say there's not just hemispheres, but there's, say, lobes. You guys know about neuroanatomical lobes? This is like frontal lobe. Here's the occipital. Maybe this is temporal. And here's the occipital. And then some other stuff. I forget all the lobes. I didn't like neuroanatomy class. But right, let's say there's 4 lobes per hemisphere. There we go. Now there's 8 by 8 thingy. So that's cool. So now we're up to 64. That's pretty good. So all this started because I asked you guys what's the right number, the true number of regions in the brain. And people said, a number of you had wonderful ideas ranging from 2 to 2 million. What I say is the right answer to that question totally depends on your sample size and what you want to do with these brains. So if your sample size was 7 billion, because you measured every single human, maybe you want to actually say that there's like 100,000 or 10,000 regions, because you can realistically estimate some of those parameters. If you have a normal study size, 20 is kind of small, 100 was a nice size study, or 1,000 is actually a pretty big study, you can realistically estimate lots of parameters. And so which model we're going to use in practice for any particular scientific question or clinical question is going to be a function of how much data we got, and also the signal to noise ratio for those data, how high quality the data are, how good our estimates are of these parameters in any given context. And so we and other people have done a bunch of stuff looking across scales, changing the number of regions because, like I said, they're all fine answers. And you can find different scales work better for different questions than other scales. Does that make sense? Does anyone have any questions about that? Nothing? Does anyone have any questions about anything else? Should we all just try to go get a free ice cream? Is that clock right? We have 15 minutes left? 2:02? Okay. So, all right, what do you guys want to do now? We'll take a vote. Just I like votes. What is this, democracy? So I could tell you how to make a slightly more general model that would be useful for other contexts. I can tell you more about other things I do in my research. I can just answer random questions about life, the universe, and everything. What do you guys want? It's the last 15 minutes of class for the year. You guys should choose what happens. Yeah?
I'd like to hear about your research.
My research is a vote. Any other votes? Yeah?
Like the universe and everything.
Okay, that one's easy. Anything else?
Life advice.
Life what?
Advice.
Life advice. Okay. Yeah? Option 3? Okay, so I'll do option 2 first because that's the answer is obvious. Everyone knows it, right?
42.
Yeah, okay. Yeah. So that one's simple. The question is more complicated. I'm working on the question. In the meantime, yeah, we can do, I'll do 10 minutes of life advice since that's fun. So, okay, who here, well, you're all alive as far as I can tell, so that's exciting. I think for me, I feel like I'm pretty successful. Like, I'm faculty in the best department in the world. That's my feeling about it. I don't know how you guys feel about it. So that's pretty cool. And I have a family and I love them. And I have a 15-month-old baby as of today. He's the best thing ever, so that's great. Yesterday he learned how to throw rocks and walk backwards. A couple days ago he spun. I think I told you guys that already, but yeah. So anyway, my belief is that the key to this success is to find what you love and then do it as much of your time as possible. And I said that last year to a class, and then people said, well, how do you find what you love? And then the answer to that is you go sit on top of a mountain by yourself for a while and see what you really want to do. And if it's play video games, then I encourage you to play video games your whole life. If it's something else, then it's something else. But I think the key, and lots of people agree with me, I've read probably 100 books on this topic at this point, is to find stuff that you love and do it as much as possible. And there are obviously constraints, like people have families, you need to pay for stuff if you want, like food and shelter. But some of it's not really that expensive. There's ways of doing it. Like the year I graduated college, I was basically homeless. I slept at the lab, slept on the roof of various buildings. I was at WashU at the time. I didn't pay rent, which made things cheap. I didn't have a car. I don't think I had a phone. I think I spent like $8,000 that year, which I don't know if you guys know how little that is to spend in a year, but it's not very much. And I was very happy, and I ate food every day, and that was great. So you can do it. It's harder when you have kids because they're hungry and stuff, but they don't eat that much. I mean, my son's name is Lion. He eats like a few hundred calories a day. Like, that's not such a big deal. Most of it comes from my wife. She's breastfeeding him, so just have to feed her, and she'll eat whatever because she's breastfeeding. So So my first note of advice is to try to find what you love, and try to do that as much as possible. And to find it, I really do recommend sitting somewhere quietly for a long time without anybody else around, without any technology, and just, you don't have to think about what you want to do, but you'll feel a desire to do stuff. And that might be the answer of what you really want to do. The other thing is, it's useful, I think, for lots of people if the thing that you end up doing is relevant to other people. Like playing video games is really fun. Like when I was in college, I think freshman year I probably played about, I don't know, maybe 60 hours of video games a week. I don't know, is there still James Bond video games? Is that a thing anymore? Not really.
GoldenEye's still good.
GoldenEye?
I mean, it's still good.
It's still good? Yeah, so we played GoldenEye in our dorm every day for a solid 10 hours a day, I'd say. Circle strafing, we killed everybody. It was awesome. But it turns out it's not that fulfilling. So Viktor Frankl is this dude. He was a Holocaust survivor and a psychologist. And he wrote a book called Man's Search for Meaning. And what he says is that, well, it should really be called Human Search for Meaning, I guess, now. But what he says is everybody likes meaning in life. And when he was sitting in the concentration camp being tortured by the Nazis, he realized something, which is that he had intellectual liberty. And he could think about whatever he wanted while they were torturing him. And the Nazi who was torturing him did not. That person basically got brainwashed into thinking that what he or she should be doing is torturing this dude. And that made him happy because he realized he had freedom, and this other person did not. And I think when I read that, I was pretty taken by that because we all kind of have intellectual freedom. I mean, this dude had it when he was being tortured, which is super impressive. Like if I have a paper cut, it's hard for me to think about anything else. But the point is, we really do have intellectual freedom. No one can stop you from thinking about whatever you want. It doesn't matter really what they're doing. You're in control of that. And so with this intellectual freedom, we get to express ourselves in lots of different ways. And it doesn't matter what job you have. You can still do it. So there's another book by this Czech psychologist called Flo, where he talks a similar thing about how everybody wants meaning in life. But what he says is, and there's a bunch of evidence about this, that just kind of ping people throughout the day randomly and kind of assess how they're feeling. People are kind of feel the best about their place in the universe when they're doing something proactively that's engaging their mind and benefiting other people in the world somehow. And this is true whether you're on a factory floor doing the most mundane job in the world or if you're a heart surgeon saving people's lives. The people who work on the factory floors who are happy, every day they try to do it a little bit better, go a little bit faster, have a little bit fewer errors, and they find meaning in that, and then they're really happy, even though that job totally sucks, obviously. And the people who are just doing cardiovascular surgery, saving people's lives, tend to not be quite as happy. So in your search for finding what you love, I would also encourage you to find something that is meaningful, not just to you, but connects you to other people also, because that seems to be lastingly meaningful to you. That's my life advice. Does anyone have any questions about that? Is anyone doing it yet? I also encourage you to study theoretical neuroscience. That's what I do. No, I actually encourage you not to do that. I mean, do that if you want. But lots of people give life advice. I found when I was growing up, almost everyone I talked to told me that I should be a doctor. Then I realized that they were all doctors. And what they meant was that they should be a doctor, which I guess is true because they were doctors. But it's really hard to give advice to people about specific things. And I find most of the time people are really projecting. So like, even when I said go sit on the mountain quietly, that's how I did it. That might not be how you would find it. So I just caution you against listening to people who give you life advice because it's really hard to do well. And we're basically just telling you what worked for us, and different things might work well for you. Yeah?
Which mountain did you go on?
Oh, I go on mountains a lot. So when I was a junior in college, summer leading into senior year, I lived in a small 2-bedroom house with 25 of my closest friends in Boulder. And because of that, we went camping a lot. So in Boulder, there's like 14ers all over the place. So we went hiking pretty much every weekend. Most weekends were about 4 or 5 days long. So there were a lot of mountains to be sitting on. In Baltimore, there's fewer mountains than in Boulder, but there's plenty of quiet places all over the world. Baltimore has them too. Yeah, even campus has some nice quiet places. Anything else? We should probably go try to get ice cream now, don't you think? Okay, let's go.